Perfect. There you go. Okay, I'm gonna share basically my slides. I don't think I would open it. Sorry, one second. Should be here. Yeah. Can just someone, one of you guys, let me know if you guys are able to see my screen?
Yes,
Yes, we can see it.
Thank you. Thank you so much, guys. Okay, guys, again, so I took this lecture. This is a lecture that my basically one of my supervisors gives, usually Vincenzo. Vince is a professor in biological drugs development. He actually has founded now two companies, basically Valo Therapeutics, which I'll be talking about today a bit. Now also Globevac, and now basically me and him together, we're gonna be also founding basically our own company, AdCab. So he is a, let's say, ranked top five best, basically, uh, virologists basically in the world. Really, he's really well known in this field. So I really like to take his, let's say, teachings and then give it on to you. And of course, I added a bit of my flavor. That's why I added my name in there towards basically what he's what's been doing in this field. So again, guys, really take this lecture purely as how we're thinking towards creating a therapy. This is really going to help you for the essay. Basically, to think, okay, how can we develop a therapy, or what's the best therapy, or how do we think about treating cancer when it comes to immunotherapy? So the content today is that we're going to talk about what immunotherapy is, what is really required for anti-tumor immunity, and then we're going to give you different examples of cancer immunotherapies, which you can put them into, let's say, three categories: passive immunotherapy, active, and this is basically a, let's say, a new kind of type that Vince kind of came up with, which he called immunoviral therapy. And we'll be talking about all these different examples. So today's it's going to be a lot about different drugs in the clinic. Today it's going to be very more therapeutic based. So this is maybe a recap of what we said last time we were had this class. So this is about again the cancer immunotherapy, let's say wheel. So how does the immune system is able to recognize a cancer? So imagine this is your tumor, right? All over here. When you have, let's say, a death of a tumor, either by chemotherapy or by sudden apoptosis or something that's happened, of course, this is going to release antigens shown here in these little red balls. Dendritic cells that are in the vicinity, what's going to happen is that they're going to come pick this up, they're going to process it and put it on their MHCs. They're going to go all the way to the lymph node where you have all your naive T cells there, and specifically CD8 T cells. Because remember, basically, most of this is basically going to be, most of these are have MHC1, so you want to take more of a CD8 perspective. So these antigens will present your CD8 T cells. They're going to basically kind of teach them, tell them like, hey, start to proliferate and start to recognize anything with that antigen, which comes from the tumor. They're going to basically proliferate, and then they're going to go in traffic all the way to the tumor, and they're going to recognize your tumor cells. So your tumor cells, as we said, these are nucleated cells. They do not have MHC2. They only have MHC1. So the majority is MHC1, basically, what they have. So it's mostly CD8 T cells that they're going to be able to recognize them. And once they recognize, they start to kill. And again, the more killing, the more release of antigens, and the more cycle you get. So this is why we call this a cycle, because the more death, the more release of antigens you have. And this was actually done by Chen and Mellman, so Ida Mellman. These are two very big scientists. They used to work in a very famous company, not Genentech. Genentech in La Jolla, California. Now, Genentech, basically, which is kind of subly owned by Roche, basically took what happened there technically is they fired all the people with cancer, specifically Ira Mellman, who was the pioneer there, because they decided not to anymore invest in cancer. But anyway, these two were the ones that developed this. And from what I know, because I have met Chen in one of the conferences, and he did tell me that him and Ira Mellman actually came up with this in a bar. They were just sitting down and they literally used a tissue to write this whole immunity cycle, which now we're using everywhere, which is actually the truth. This is basically what you need really All these are the all necessary steps that you need to actually induce a immune response against your tumor. So our therapies are trying to technically enhance one of these steps. So when it comes to immunotherapy, there's different types. There's active, passive, and then there's immunotherapy. I'm gonna just right now really concentrate on active and passive. So active immunotherapy, it basically means it's in some sort of way you're activating your immune system to induce an basically an immune cycle. So it's active. So these are basically could be either, let's say, drugs that activate your B cells and T cells to get activated and start producing antibodies or to start actually creating a CD8 T cell response. So these could be specific or non-specific. These could be non-specific like adjuvants. Adjuvants are things that activate your, let's say, dendritic cells to start doing more antigen processing. Or these could be specific. These could be, for example, vaccines. These could be something that are specifically activating your immune system to kill something specifically. Non-specific, no. You're just activating worldwide dendritic cells, and whatever the dendritic cell picks up, it's going to pick up. Hence, it's non-specific. Passive is it basically means the opposite of active. It's basically these things do not activate technically your own immune cell to kill. No, these are basically drugs that you go and they literally go and go straight to the cancer and kill. These basically could be literally you expand your own T cells outside and then you inject them, they go straight and kill. These could be antibodies that they literally you inject antibodies and they go straight to the tumor. Hence, it's passive because you're not, let's say, activating your immune cycle. You're not activating your, let's say, T cells and B cells in your immune system to do the killing. Active in this kind of sense, no, you are giving certain drugs basically that will activate your B or T cells or dendritic cells to basically create an immune response. So I'll give you examples. For example, let's start with active. So when it comes to active, the steps that you really mostly are trying to do is mostly around here. You're trying, for example, this is more the non-specific kind of thing it can be. In a sense, you're activating dendritic cells to do more antigen presentation, or you're making dendritic cells come more to the tumor. The tumor, we sometimes call it a desert in a sense that there's not a lot of immune cells, and they do it on purpose. They create this very immunosuppressive environment. So what you can technically do is you can inject a lot of molecules that will enhance, let's say, your immune system to come here, and one of them could be dendritic cells to do antigen presentation. Or you can actually add certain adjuvants, certain molecules that make dendritic cells much more active in antigen presentation. They start to upregulate MHC1, capacin, all these different types of molecules related to processing. Or you can basically also make them dendritic cell. You can actually inject dendritic cells into a patient. And these dendritic cells are engineered to become much more aggressive in going to the lymph node, for example. There's so many ways that I can come up with, and I'll give you examples. Or you can basically, in some sort of way, give certain vaccinations or something like that here to kind of enhance your T cell. So let's start with a non-specific. These are adjuvants. So adjuvants are technically molecules that induce your basically an activation. This activation is usually non-specific. So technically you're activating mostly your innate immune cells. Basically, what you're trying to do is mostly here, you're trying to activate your dendritic cells because a inactive dendritic cell is not really going to do a lot of antigen presentation. They're not going to present uh, antigens on their MHC, but when they're activated, they do much more. So one thing that you can technically do is add actually toxins. Toxins are very good molecules that are able to activate your immune system very, very well. Of course, these toxins are not fully, let's say, capable of killing us. They are, let's say, attenuated, meaning that you've removed a bit of their killing capabilities. Another one that is very used actually in the clinic is BCG. So BCG, we call it Bacillus calmette-guerin. This is a vaccine, but this is a very old vaccine that basically is mostly like since the 1800s. It's a vaccine used to prevent tuberculosis, TB. So tuberculosis is a very nasty disease that is usually caused by Mycobacterium tuberculosis. It's a bacteria that causes a lot of pneumonia, and it persists in our lungs for basically a lifetime. So we developed basically a vaccine which is called BCG, which is technically the bacteria, this Mycobacterium tuberculosis, but it's passed in potato skins more than 1,000 times. Why do you do that? Is simply because now the mycobacterium is so attenuated. It's so basically, let's put it this way, non-functional. It's barely alive. It's, let's say, kind of you've really crippled the hell out of the bacteria. So when you inject it in a human being, that bacterium can barely infect. It's just barely just it's just there. It's semi-alive. That can be per se be a vaccine because that whole bacteria is able to activate the immune response and basically, in some sort of way, create a dendritic cell to come up, pick it up, and present antigens to your T cells. So this is basically, it was a very old vaccine for tuberculosis. The thing about it is that it's not super effective to the point that very little, let's say, countries are now using it. I think Lebanon people don't actually vaccinate. It's not in the program. I know in Italy or Spain it's not. In some basically countries that are very, let's say, kind of have an old school mentality, like Germany, for example, they have it. But BCG is not really in the vaccinating kind of program. But this physician, especially, sorry, I have a question. That I read that they're trying to activate modify neutrophils under this non-specific approach because they're initial responders. Is this true? Yes, Miriam, this is very, very true. They're using BCG vaccines because BCG vaccine can activate very well your innate immune system. One of them is specifically your dendritic cells, but now new studies have come out that they actually activate your neutrophils extremely well. They do this in a very very weird way that I'm not going to be speaking about because it's a bit advanced, but they do something called trained immunity. They train the neutrophils to become much more responsive towards anything, towards viruses, towards parasites, but even towards cancer. So it's very cool how BCG is now being used. So in this context, people were using BCG to activate your innate immune cells, like neutrophils, for example, like Miriam was kind of saying, but also to activate your dendritic cells. You make your dendritic cells more awake, they start to do more antigen presentation. And this has actually been now accepted in bladder cancer. So now what people do, basically physicians, when people have bladder cancer, specifically it's a form that's called invasive muscle bladder cancer. It's a cancer that's now invading the muscles near your bladder. This is very metastatic kind of type of bladder cancer. You inject BCG straight into it, and basically, this really helps you activate your dendritic cells to create a cancer immunotherapy kind of circle. So you're trying to get your dendritic cells much more active. Another thing that you can technically do is basically more actively is actually create, get dendritic cells. What you can technically do is you can actually take dendritic cells and inject them. This was basically actually something that won the Nobel Prize. I think it was in 2008 by Ralph Stein. So what he saw that what you can do with patients is you can take a whole dendritic cells, expand them a lot, let's say outside, ex vivo, and then you inject it into a patient. So now the patient has a lot of dendritic cells, which will then go into your tumor and start to present. So that's basically one way. So I'll show you basically here. So either what you can technically do is two things. You can take dendritic cells and you can basically feed them with the tumor coming from the patient. And what happens is basically now these dendritic cells are going to eat up these tumor cells and they're going to present whatever. Or what you can technically do is you can take dendritic cells and feed them with specifically the tumor peptide that you want. So if you know what peptide coming from the tumor you want to vaccinate it against, then you feed that. It becomes much more easier. If you don't know, let's imagine that you don't know really the tumor peptide, you just give the whole cancer and let the dendritic cell decide whatever tumor peptide it wants. It will decide by itself. You basically expand these dendritic cells, you feed them with whatever peptides you want, either peptides or whole cancer, and then you inject them again back into your basically human. And this is called peptide-based vaccine therapy or basically dendritic cell also therapy. So you can have dendritic cell peptide pulsed or dendritic cell tumor pulse therapy in some sort of way. And what you're trying to do is you're trying to create specific T cell responses. So let's imagine here you have these sorry, these red tumor peptides or this green tumor peptides, well, you're creating basically T cells against specifically against your basically your red antigens or your green antigens. Actually, the person that discovered this, Ralph Stein, this is a really nice story. Ralph Stein discovered these kind of, let's say, new type of therapies. He actually got diagnosed with pancreatic cancer, and he was such a huge believer in his own therapy that he started to actually inject his own self with dendritic cell therapy. He started to take actually his own dendritic cells, expand them outside his body into huge numbers. And then he used to feed them with his own tumor. So he used to take small biopsies from his pancreatic cancer, feed them, and then inject them back to himself. And believe it or not, Ralph Stein lived, I think, 1 or 2 years extra. And with pancreatic cancer, that's very rare. Usually, pancreatic cancer has an overall survival of less than 5%, or even it's around 3%, and your medium survival rate is about, I think, between 5 to 8 months. So he managed to prolong his life through that. He was awarded the Nobel Prize. He got so unlucky that he actually died, I think, a couple of days before the him getting the Nobel Prize. And there's a rule that you're not allowed to give Nobel Prizes to dead scientists. So his wife lied to the committee, actually. She said to the committee that he's alive and he's still actually in order to get the prize. So she went to Sweden, to Stockholm to receive the prize. And then 2, 3 days later, she announced his death. Everyone knew. And I think everyone turned a blind eye because he deserved it. He deserved it at the end of the day. So dendritic cell therapy is a huge, let's say, kind of therapy or wider therapy that's going into the clinic. One of them that's actually now used for prostate cancer is called Provogene, Cipocel T. It's literally dendritic cells that you feed it actually with tumor peptides coming from prostate cancer. They use it. It's quite good. Now there's currently, let's say, if you go right now, there's a very nice website called clinicaltrials.gov. It's a very good website from the US that provides you every single clinical trial that's going on in the world. And if you go right there and you basically, let's say, put the treatment dendritic cells, you have around 631 dendritic cell basically clinical trials currently ongoing. It's a huge avenue that people are testing. But to say, in my opinion, it's a failing one, in my opinion. It's not really working as well as we thought, to the point that despite so many studies, there's only one, let's say, one of them that's actually really approved, technically. So another type of basically kind of therapy that's similar to your dendritic cells are your basically vaccines, because technically your dendritic cells that are now basically post with your peptides or your tumor, they're technically vaccines because they're inducing a T cell response. Well, you can do the same thing, but by using different carriers. You can use basically viruses expressing your, let's say, tumor antigens. You could use basically specifically antigens, peptides. You can inject peptides directly. They're going to be taken up by the dendritic cells, or you can use mRNA vaccines or DNA vaccines. And I want to talk about mRNA vaccines because this is a vaccine that I'm very sure every one of you guys have heard. This is the one that we basically use for, let's say, we use it for COVID, but now this is becoming a huge thing for cancer. So what are mRNA vaccines? They're technically these lipid nanoparticles. These are nanoparticles made of lipids that technically have an mRNA. And what the mRNA has, it encodes for a peptide, depending on what that peptide you want to vaccinate for. So what you can technically do is you inject this, and you specifically inject this in your muscle cells, because what's going to happen is that this mRNA vaccine, this mRNA is going to go into your muscle, and the muscle cells are going to start producing a lot of that peptide, and they're going to be presented or taken up by dendritic cells. Dendritic cells are going to take that peptides, and they're going to basically get presented to your CD8 and CD4 T cells, which they're trying to basically kind of teach them what to kill. So this is basically what your mRNA vaccine technically has. Very importantly, you have these coding sequence. This coding sequence basically is your peptide. Whatever peptide that you want, or you're trying to basically kind of vaccinate against, this is the sequence that you take. And then you have these untranslated regions, because usually this coding sequence is very small. We're talking about, let's imagine, 100 base pairs. That's very small. So what you can technically do is you can, to make this mRNA vaccine a bit much more bigger, because the bigger it is, the more stable it becomes. You add these untranslated regions, which are basically just blank space. They're just regions that are not never going to be translated, and they're just there to make the mRNA molecule much bigger. But mRNA basically is very unstable compared to DNA, because DNA is double-stranded, mRNA is single-stranded, so it's not very stable. So to make it very stable, you add these cap structures. So it's a cap over here at the 5 prime, and you put this poly A tail in 3 prime, and this really helps this whole mRNA sequence from not being degraded. Because in our system, we have a lot of you can degrade from the 5 prime or the 3 prime basically areas. So to prevent that, you put a cap structure or a poly A tail and the 3 prime. And this is basically your mRNA vaccine. It's very simple. So it's just knowing what peptide you want to sequence with. So this remains constant. The only thing that basically changes is this coding sequence, and depends on what peptide that you want to, let's say, vaccinate against. But there was a very cool, let's say, interesting thing that we saw. We used to see that if we produced this mRNA structure in a test tube, this would actually not be working really well, because what would happen is that when let's say, imagine we created this mRNA vaccine in the test tube, when we actually injected it in a human being, we would not get any vaccine response, because what would happen is you would actually create such a huge inflammatory response that the cell that has the mRNA would basically destroy it before presenting any antigen. But if we created this mRNA vaccine, or this mRNA, let's say, strand, inside a mammalian cell, this would be fine. You would not get this massive inflammatory response, and basically the cell would create so much of these peptides that would be able to be taken up by their dendritic cells. But why? Why is that mRNA produced in a test tube was getting an inflammatory response that was killing the cells producing it, and the one produced in a eukaryotic cell or a mammalian cell was not? And this was actually an observation done by Carico and Weisman. So what they saw was that in a test tube, the uridine that was put in the mRNA was not modified. And this was basically seen from our immune system as very immunogenic. It was seen as very foreign, and our immune response was trying to destroy anything that had that uridine. In mammalian cells, this uridine that we have is usually modified. So you have basically this kind of, let's say, modification over here with a nitrogen. This is usually a carbon. This is now basically it's modified, and we call this pseudouridine. And this is basically less immunogenic. It doesn't create immune response. It's how basically this mRNA can basically persist in our body. And this is something that they gave them basically the Nobel Prize. And this really, let's say, pushed mRNA vaccination to the full limit. So this is just an example of how good mRNA vaccine. This is a paper in 2023, basically by Rojas in Nature. So Nature is one of the most top best journals you can ever publish in. So anything you see in Nature, it means it's of the highest quality technically. So what they did here, this is basically they actually was the company. This was the company that did the mRNA vaccine, which was BioNTech. So BioNTech, what they did is they actually got something of basically 34 pancreatic cancer patients. And of there's 34, basically they managed to take 16. So 16 of them basically saw that they were able to, let's say, create an mRNA vaccine. So from these 16 people, they literally took a piece of their tumor. They took the RNA from this tumor, and they started to see what are the new antigens? What are the tumor peptides that have mutations? So remember, tumor antigen, these new antigens are technically tumor peptides that have a mutation and are very new. They are not found anywhere else but in the tumor. This is the holy grail. They have very sophisticated programs to detect this. And they created this mRNA vaccines. So the mRNA basically encoded for the new antigens they found. And this is very personalized, meaning that all 16 of these patients had different vaccines. They had very different mRNA depending on what new antigens they found. And this is the new wave of therapy. It's called personalized cancer immunotherapy, meaning that your therapy is not something that is just given, that it's like a one therapy given to everyone. No, you are designed a therapy just for you based on your tumor. So they created basically these vaccines, and then this is basically what they did. They basically gave 8 doses of this mRNA vaccine. And then they give this FOLFIRINOX, which is technically a chemotherapy regimen. So don't worry about this. This is just to show, this is the C and D. It's just to show that there was not a lot of basically adverse effects, nothing more of the, out of the ordinary. So here, this is the important part, the E. So here they were looking at basically, did these patients have a immune response or not? So did they have T cells that were recognizing this mRNA that they were vaccinating against? So here, basically, they saw that 8 out of the 16, 8 of them had a response. So they had a T cell that was actually, responding towards the peptide or the mRNA that were being vaccinated against, and 8 of them didn't. And this is to show you that it's not 100% true that the vaccine will create a T cell response. So selecting the right peptide is extremely hard. It's actually one of the most hardest things ever, because here in the 16 people, only half of them actually had the correct, let's say, mRNA selected for them. So 50% basically had a T cell response and 50% didn't. And that coincided actually with people that survived or not. The people that had a T cell response survived, while the ones that basically didn't have a T cell response did not survive. They died actually 5, 6 months later on. The people that actually had a T cell response, they're still living. 5 years later, Rojas published another paper showing that these people are still living and that their T cell response was still ongoing. This is how good this vaccine is. This was unheard of in pancreatic cancer. You don't see this. And they got so encouraged with these results that they now have basically now are doing the same thing with another aggressive cancer called triple negative breast cancer. It's a subtype of breast cancer that is extremely aggressive. It usually you don't have a lot of therapies for that. And again, they're seeing very good results with that. So this mRNA vaccination is extremely potent. So these are basically the more, let's say, active, basically therapies, the ones that you're trying to create a vaccine-like response, either by non-specific, by injecting toxins or a whole BCG vaccine to increase your dendritic cells or your neutrophils to create, let's say, a T cell response, or you're doing it specifically by creating a vaccine, by giving a vaccine. This could be dendritic cells with peptides ready to present to T cells. This could be mRNA vaccines. This could be pure peptides straight into your dendritic cells and your system. A lot of things can be. Now let's talk about passive. Passive, again, is where you're trying not to basically create immune response. You're creating a drug that is going straight to a tumor to kill. So what you're trying to do is these therapies are going straight to your tumor, binding to your tumor, and they're trying to kill the tumor. And they're doing this by making, for example, T cells recognize the tumor, or by themselves activating, let's say, innate immune cells to come and kill, like macrophages or dendritic cells or whatever. We'll talk about all different examples. So one of them, technically, is called adoptive T cell therapy. So this is a very cool type of therapy in where people start to recognize, okay, this whole cycle, we're basically trying to end up with having a T cell coming in and killing. But what about if we just increase the amount of T cells? How about we make T cells, a huge army of them, and inject them into the patient to kill? And that was called adoptive T cell therapy. We also call this TIL therapy, basically T cell infusion therapy. So what you do is you take this is let's imagine this is your patient. You take the T cell, basically let's say blood. You take the T cells from the blood. Basically you can able to actually kind of separate them. And then what you do is literally you activate them and you expand them. So let's imagine here you have, let's say, 1 million T cells. You can expand them to 1 billion. T cells are so easy to expand. I do this on a daily basis. It's very easy. You just add a cocktail of cytokines and they start expanding like there's no tomorrow. So you suddenly from let's say, million, you're increasing them by 3 logs. And then you infuse it back to the patient. So now this patient has a bigger army, 3 logs more of what they originally had. And now you have a big army ready to go, your T cells. This has been shown to work, but not a lot. It's super expensive and not that effective because what happens in your tumor, as we know, the tumor can downregulate your MHC. It can really start not producing any more MHCs. So your T cells are there. You have an army of T cells, but great. They're not going to recognize it. There's no MHC. So you've just upregulated them for nothing, technically. Another thing is that around your tumors, you can have a huge immunosuppressive, basically, array. So your tumor can bring Tregs, they can bring myeloid-derived suppressor cells, all these cells that are literally stopping your T cells from doing anything. So you have an army, but that's being stopped. So it doesn't really do much. So, and also what this also sometimes means that you need to basically, when you want to expand T cells, this means that the patient has to have T cells that are actually by themselves, let's say, recognizing the cancer. Because you can expand 1 million, let's say, T cells to 1 billion, but all of them are let's say, recognizing microorganisms, viruses, fungi, bacteria, but none of them is recognizing a tumor cell. So you've done nothing. So that's also an issue people are seeing. So what people started to do is like, okay, let's make an army of T cells, but let's make them more educated, or let's give them a superpower to make them recognize the tumor much, much better. And they developed a new, basically, kind of T cell that we call them CAR T cells. CAR is basically means chimeric antigen receptor. So as we know, if we go back into here, basically, T cells, they recognize tumors by MHC with a peptide. Scientists said, how can we bypass this? Because this is a very complicated, actually, system. Because that means we need to we need to have the perfect TCR, and we need to select the proper TCRs and all these things. How can we bypass this? So what they did is they took an antibody. Antibodies are very quite nice because you can take any antibody against any antigen. So let's imagine you have, let's say, B cells. Well, we know B cells, malignant B cells, they express CD20. Well, you can create an antibody against CD20. And we know that the antibodies, the part that binds to CD20, is the variable regions. We take those variable regions that are binding to CD20 and we connect them to the signaling pathway of the TCR. So you have the TCR here. This is your TCR that recognizes the peptide. And next to it, you have the CD3. It has basically this, we call this region the signal transduction domain of the T cell receptor, the CD3F. This is the signal. So once this basically, let's say TCR recognizes the antigen, it induces or makes this part active, and it causes basically a signal in transduction to make the T cell start to kill. So we took this part and we added it towards this variable region. So now you created something called a CAR, a chimeric antigen receptor, a CAR. So what does this mean? That now we have a novel receptor that is recognizing basically an antigen using this antibody part. It's not recognizing it through an MHC with a peptide. No. Now it's recognizing it purely based on what's binding to. When this is activated or binds to its antigen, it's going to activate the signal transduction here, and it's going to make your T cells start to kill. This is basically now you've made a T cell now able to kill MHC independent. So it doesn't matter that your tumor downregulates your MHC. We don't care about that because now we have this receptor that's able to recognize the antigen found. Or this could be a receptor. This could be, for example, CD20 on your B cells. This could be HER2 on basically breast cancer. This could be, for example, colon cancer express a lot of EGFR receptors. This could be EGFR. This could be any receptor on the extracellular matrix that basically this is basically being able to bind to. So remember, this basically antibodies will only bind to basically proteins on the extracellular matrix. Antibodies cannot pass inside the cell. So this is very limited to what the tumor cell is expressing on the outside. And this is called a CAR T cell receptor. Do they modify it with a VDJ coupling? Technically, you can modify it. Yes, basically, of course, it is all about modifying your, let's say, variable region depending what you want it, and then having a signal transduction. It's more these kind of things that you're trying to modify, and I'll definitely tell you more what the modifications are technically there. So this is, for example, I'm going to show you how what basically happens. So a T cell, as we said, a T cell gets activated usually by the MHC presenting with an antigen. Usually that's the way how it's done. But now when you add a CAR T cell, it's completely switched because we don't care anymore about this MHC with the antigen. No. Now that you have basically this CAR, there's basically variable region coming from the antibodies binding to a tumor antigen that's expressed on the membrane of your tumor cell. This basically is able to get activated. This basically binds. This activates your CD3F, that's causing a signaling, which is causing the T cell to get very activated and release its perforating granzymes to do apoptosis. So now you went from a MHC dependent killing to an MHC independent killing. And Miriam, to basically, let's say, answer your questions, the VGA coupling, you don't really do this a lot. What you do much more in a T cell is you're trying to make it much more active. How do you do this more active? So the first generation was purely just having the CD3F, so this literally transduction domain. This was good, but it wasn't very activated. It didn't get activated that much. So to increase the activation, they kept adding co-stimulatory molecules. So to this CD3F, they added the co-stimulation of CD28 or 4-1BB. CD28, guys, remember, these are the co-stimulation molecules. They also have a transduction pathway. So you just add it to here. The third generation, you added another, basically, let's say, domain that gets basically more activated, and the fourth generation is even more. So you're just increasing much more signaling domains to make this CAR T cell much more active. So this part binds, and this does it very well. But the lower part, to get activated, apparently they started to see that they need much more domains to get this very active and make a T cell much more activated. So the fourth generation is literally having four different types of domains coming from the CD3, from 4-1BB, for CD28, or different types of signaling domains that a T cell uses to activate their pathways. So again, this is just to show you, again, the more concept of CAR T cells. The main way of a T cell, how it kills the tumor cell, is through MHC and a tumor peptide. And we know that tumor cells, they really downregulate a lot of MHC1s, and having the proper TCR basically bind to the tumor peptide might be very little. There can be literally 3 or 4 clones in your body, so you have very little amount of them. So what you can do is you can take T cells and you can basically make them express this new CAR T cell. There's now, let's say, CAR, that is, you have this signal, basically these variable chains binding to a tumor antigen that have this signaling domains, either from the CD3, F, or coming from CD28, or from 4-1BB, different ones. So this is basically what would happen, let's say in the clinic. You would take T cells, basically, or let's say the blood from a patient. You would increase the amount of T cells, you would, let's expand them, and then you would insert basically the DNA encoding for this CAR inside this T cell, because now this DNA will actually give this T cell the ability to produce CAR, so this chimeric antigen receptor. How you insert this gene, you can do many ways. You can usually people what they do is they take lentiviruses, certain viruses that these viruses do is they infect the T cell and they basically incorporate this gene inside the genome of this T cell. And now these T cells are expressing this CAR very well. You expand them, you create a huge, literally millions or hundreds of millions of them, and you fuse them back into the patient. And now the patient has a T cell that has all these CARs that are able to recognize the cancer cell. And you're basically now giving, let's say, your own T cell a superpower. You're giving them a CAR receptor. This works extremely well. There's many CAR T cells right now. Let's say the biggest blocking busters that are most probably going to come up in the next 5 to 10 years will be CAR T cells. There is thousands of clinical trials ongoing on CAR T cells, and there's I think like a couple of them that are now accepted. I think mostly for lymphomas that are being accepted right now. Very great. But the issue with CAR T cells, it's extremely expensive. We're talking about millions. We really are talking about basically millions per year coming from one patient. For certain countries, for example, in Europe, you have very good systems where you don't pay much or barely anything. But in places, for example, like in the US or in basically Lebanon, where sometimes people, their insurances will not cover it, they're not able to receive this kind of treatments. And this can be a huge issue. Of course, the thing about why this is expensive is because this whole process takes about a month, and you need experts, you need clean rooms. It's extremely very personalized because the T cells that you're taking, they have to be coming from the patient. You're not allowed to take a T cell from someone else because let's imagine if I go and take T cells from a random person, I inject it to myself, I'm creating an autoimmune response because the T cells that this other person have different MHCs, they're recognizing different things, they can start killing me. So this has to come from basically the same person because the T cell has to have the same MHC as we do. Because again, if it doesn't, this can cause autoimmunity or basically a lot of different rejection transplants. Like it's a whole transplant rejection happening. So scientists, of course, started to see like this is such a great tool, but it's so expensive. How can we create a therapy that we have an basically off-the-shelf product, basically a product that we can take from the shelf and then basically inject it to every single person. One product serving all to make this much, much more cheaper. And they came up with something called a BiTE. We call them bispecific T cell engager. And I love the name BiTE because it really is like a bite. So a BiTE really works very, very similar to a CAR T cell. So what it technically is, it's combining two antibodies together. So let's imagine here you have two antibodies, one antibody that binds to your T cells, specifically to CD3. So all T cells express CD3. And then you have your tumor cell. Basically, you have an antibody binding basically to a receptor on your tumor cell. What you can do, you take the variable region from the CD3 T cell and the variable region from this now antibody binding to your tumor, and you combine them together. You create something called a BiTE. So this BiTE now has a variable region binding to your T cell and a variable region binding to your tumor cell. When you inject this to the patient, what's going to happen is that this CD3 part is going to bind to your T cell. This is going to bind to your tumor, and they're going to bring them so close to proximity that they're going to kill. For some reason, I do not know why. The mechanisms are still unknown, but the proximity of bringing a T cell so close to your tumor cell, thanks to this BiTE, causes the T cell to get activated and kill your tumor cell. We call this the kiss of death. You're literally making the T cell kiss your tumor cell and is allowing it to kill it, hence the kiss of death. So now you're making a T cell kill MHC independently. So the T cell is not killing through the MHC anymore. No, it's killing again through the variable region. Very similar to your CAR T cell, but it worked. But it's basically much, much more cheaper because this is a product that you can create off, and you can put it on the shelf, and you basically can use it for any patient with basically that as long as this portion is binding to the tumor cell. So let's imagine, for example, breast cancer. Breast cancer, we know specifically there's a subtype of breast cancer that they all produce HER2. HER2 is basically a receptor found on breast cancer cells, really a lot. So you can use basically a BiTE that it binds to the HER2. This binds to CD3. This now can be used for any person that actually has breast cancer expressing HER2. So this is much more cheaper and is extremely effective. A very good one that's actually in the clinic, it's called Blincyto. Blincyto, basically, it's mostly for malignant B cells, so lymphomas. So you have a part that binds to your CD3. So this is the variable region binding to your CD3. And this other one is binding to CD19. So you're making your T cell come super close to your malignant B cell that they're literally killing each other. Well, sorry, more the T cell is killing the B cell. This is basically a very cool clinical trials that have come up basically here. So these are basically people giving blinatumomab. So blinatumomab is Blincyto. It's just the antibody name of it. And these are people basically giving chemotherapy. You see that people giving this therapy have a literally increase, a massive increase, almost double. So if you look at the overall survival, people giving chemotherapy have 50% chance of living, while the people now having basically blinatumomab is 80%. So you really increased the overall survival by double. This is how good this therapy is. And honestly, now they've been doing it for so many different types of basically cancers. So the ones that we saw are mostly for lymphomas because they're CD19, so these are B cells. But these are basically used, for example, for HER2. This is basically found in breast cancer. EpCAM, basically, these are found in ascites. Basically, you also have this in HER2 for metastatic breast cancer. You have again lung with you the express EpCAM, CEA that's found in colorectal. So most of these are coming for CD3. But what you can also do is the one binding to your basically T cells, you can change it and you can have CD16. CD16 binds to NK cells. CD64 binds to macrophages. So rather than just having T cells coming in to kill, you can bring other immune cells to come also kill. So they started to realize that what we can technically do is we can bring different types of immune cells close to proximity to a tumor cell. So again, this part has to bind to any basically receptor, expressed in a tumor cell, while this part can bind to any immune cell. Because the key thing is to bring the immune cell to close proximity towards your tumor cell, and that's going to induce killing. The mechanisms are still unknown, which to me is fascinating. Yes, Miriam, tell me.
Um, can you hear me?
Yes, I can hear you.
Okay, so for B-cell lymphoma, for example, the assignment, and I noticed it here. It always depends on tumor-specific antigens, tumor-specific proteins. For example, in the mRNA vaccine, only 16 patients could like actually get the vaccine and get this
Mhm.
targeted therapy. So when we want to choose an immunotherapy, how inclusive can we be? Because it's very difficult to be inclusive.
That's a very, very good question, Miriam. And I'm gonna let you know basically on this. So basically, so this is for example, in this kind of case over here, it's CD19. Technically, when you go, because now this is approved. So now once it's approved, it has to be used on everyone. But when it comes to the mRNA vaccine, it was a phase one clinical trial. Especially with phase one clinical trial, with phase clinical trials, the issue with them is that companies are becoming super selective. So they started off with 32 patients, but they selected 16 to be vaccinated purely because these 16 most probably were very early on with their cancer and had a very good prognosis because they want these people. Because if they choose people that have a very poor prognosis, the probability of the therapy to work is very low. And if they fail, the whole therapy goes to trash. So most probably this mRNA vaccine could be working with many people. You never know. But during clinical trials, they're being extremely selective because you want to pick the best patients that will most probably work with. So that's something you need to take with a pinch of salt. And I understand from a pharmaceutical company perspective that if we are not able to select these patients, we're going to fail and we're going to lose millions of basically of dollars, or even almost a billion or something. But from a patient perspective, you're basically being selective towards the people that that really don't need it. These people can basically live on other therapies, while the people that really need it, you're not selecting them. So this is more of a clinical trial kind of perspective. Do you kind of understand what I'm trying to say?
Yes, and even in like B-cell lymphoma, I read that there's sometimes a fusion protein in the chromosomes that causes um, B-cell lymphoma, but other patients don't have it. It's not necessarily due to this fusion protein. So, um, so there's targeted therapies can only take a part of some patients to certain places to better places.
For sure. Yes. And what you're talking about is I know which one you're talking about. This fusion one, I forgot the name of it. This is mostly found in CML, in chronic myeloid leukemia. You have exactly this target, this fusion protein that creates a new signaling product that makes the B cells to go uncontrollably. They start to like uncontrollably start proliferating. So CML is lymphoma, sorry, not leukemia. And yes, it's only patients that have that fusion that you can target it. But when it comes to, for example, CD19, all lymphomas have CD19. Like B cells all have CD19. So here, yes, your patients. So whenever you're creating a therapy, you need to make sure that your therapy is able to target the tumor. So with the mRNA vaccine, there they could technically, because there it's personalized. You're creating a personalized therapy based on the mRNA they're expressing. So you're not doing a one therapy fits all. No, you're making it very tailor specific. So in the clinical trial, they just selected the people that they knew that most probably will live because they have a very good prognosis. But in this case over here, of course, you're not going to be able to use this, for example, with a leukemia, for example, a monocyte-based leukemia, because monocytes don't have CD19. So yes, it's very target specific.
Okay, thank you.
No worries. So another very, very cool thing that came up now is this is a tri-specific antibody. This is a new product coming from Sanofi. Sanofi basically said, you know what? We saw that in CAR T cells that we started to see the first and second, third generations were basically, making a T cell much more active by adding more signaling domains. Well, we can do the same thing. So what they did is they got this tri-specific antibody. So where they had one arm that binds to basically a tumor antigen. So in this case, it's CD38. So this is myeloma cell. Myeloma is cancer coming from your plasma cells, your plasma B cells. Plasma B cells do not actually have CD19 or CD20. Myeloma cells, they're downregulated. So that's why you use CD38 because CD38 is very specific towards plasma cells. So one arm is binding to CD38, while the second arm, they made it in a very, it's called click chemistry in a way that they had one arm of this here that binds to CD3 and the other one that binds to CD28. So they basically re-engineer the whole antibody to bind to three portions. One to the myeloma cell, but the other one, it binds to CD3, which causes basically the CD3F domain to get activated, but also CD28. And as we know, CD28 is a co-stimulation, so your T cell is getting double activated. So it's killing much, much more. This is now, I think, if not mistaken, in phase 2 or phase 3 clinical trials. It's showing a lot of promise. So these are basically antibodies. Of course, another type, a very, let's say, very big type of antibodies being used right now in the clinics are just antibodies. Antibodies are about binding towards targets. And how antibodies are able to kill is actually by blocking. So what you can do, as we know, tumor cells, they express a lot of receptors, growth receptors, and they need these growth receptors to grow. So an antibody can block it or can block the ligand from basically coming to your growth receptor. The antibody can bind towards basically these receptors and now suddenly signal them to actually to do apoptosis, or they can bind and they can actually induce effector cells, so like NK cells or macrophages or neutrophils to come and kill the target. Because remember, antibodies have an FC, innate immune cells have FC receptors that are able to bind and they're able to kill the target cell, or able to activate your complement system to come and kill your target cells. So I'll give you an example with B cell malignancies. So B cell malignancies, actually, by the way, is the fifth most frequent cancer in Europe. Its incidence increases steadily, and 95% of all lymphoid neoplasms, so all lymphomas, are B cells. And B cells have CD20 or CD19. So technically, we can create antibodies against CD20 or CD19. So let's imagine these are your malignant B cells, and this red triangle is CD20. You get your antibody that binds to CD20, and what can happen is you get your complement system getting activated, or basically you get your macrophages, monocytes, neutrophils, or NK cells coming in to actually kill them. Because as we know, your antibodies, your IgG antibodies, they can induce the classical pathway of complement. Complement actually starts to kill your tumors, or the antibody can bind to FC receptors found in your immune cells, and you have the death of them. We also see this a lot, for example, with breast cancer. So, as I said, with breast cancer upregulates a type of receptor called HER2. HER2 is a growth receptor, and they use it actually to really multiply quite a lot. So, a normal cell might have, let's say, 4 copies of HER2, but a breast cancer might have hundreds of copies of it. So, we can use that as a target. And we created an antibody against HER2 called trastuzumab. So trastuzumab is an antibody that binds to HER2 in specific, and it actually can do actually block the signaling of HER2. So now suddenly this breast cancer that needs HER2 to really proliferate does not receive any signaling. But it also can actually cause, of course, uh, NK cells, macrophages, neutrophils to come and kill. And trastuzumab is a very old drug. It's not a new one. It's been used now for the past, let's say, decade or two. This is one of the first clinical trials ever done. So this is basically patients having breast cancer expressing HER2. This is basically people, them receiving chemotherapy. And this is basically showing here trastuzumab plus chemotherapy. And you saw a 25% increase of overall survival. And this was a very old clinical trial. And one of my professors actually told me that he was in the conference when they presented this data. And apparently, people started to cry out of joy because they started to see that we can actually start target cancer and have good results. Because back in the day, results were very devastating. That this 25% increase, as little as it is, it was a huge boost for the cancer therapy kind of world. Another one that is very used right now in therapy is checkpoint inhibitor therapy. And these are antibodies that help T cells recognize your tumor. This is something that has actually won. Let's get a mixture. Isn't chemotherapy immunosuppressive? How these combination therapy work? William, that's again another excellent question that you're asking there. Yes, chemotherapy is actually immunosuppressive to the point that the regimen of chemotherapy nowadays has been now trying to basically mitigate it. So chemotherapy still does work. Like we cannot your new therapies that are going to come out are never going to change chemotherapy. Chemotherapy will only be reduced, and it will be reduced to reduce the basically side effects, but also to reduce immunosuppression. But they just work. They work in killing cancer cells, and the more death you have, the better. So if you have more than one therapy killing, it's better because what we're seeing is that it's never this thing of having one therapy ever works. Usually it's the combination of therapies that work. So chemotherapy will always be there. We'll just reduce the amount of it to not make it super immunosuppressive. But yes, what you asked is extremely important. Another good therapy that's really, really now, let's say the biggest cancer immunotherapy in the clinic right now is checkpoint inhibitor therapy. And this is something that James Allison and Tasuku Honyo actually discovered. They gave them the Nobel Prize in 2018. So as we remember, this is something that goes back to central tolerance. We spoke about this a lot. So we know that T cells, to get actually activated, they need three signals. Signal number one coming from MHC and your peptide. So that's signal number one. Signal number two is the co-stimulation, so the CD28 and the CD80. And the third one is that's not drawn here, is your interleukin 2. But what happens sometimes is that your dendritic cell can have something like CD4. Sorry, CTLA4. Well, CTLA4, so basically here, CTLA4 actually, sorry, this here, CTLA4 basically does the opposite of your CD8 and 86. And what it does, it literally will inhibit, will rather than a positive signal, a negative signal. And this can make your T cell actually go down. Tregs have this, and they start to produce a lot of CTLA4. Another thing that's very similar is tissues can express a very similar, let's say, receptor like CTLA4 that's called PDL1 or PDL2, but specifically more PDL1. So PDL1 can increase, and this can bind to PD1 on T cells, and this can cause T cells now to suddenly not work anymore because this is a negative signal and cause T cells to shut down, to go into anarchy. One thing that people started to see, specifically Tasuko and James Allison, Tasuko saw that cancer cells were upregulating PDL1 like crazy, quite a lot of it. So what was happening is that even though the patient had T cells that were recognizing the tumor, they were not able to kill. So even though that their TCRs are binding to the cancer through the MHC and peptide, they were not killing because the tumors had so much PDL1, which was binding to PD1 and causing these T cells to stop. But actually, James Allison, he also saw something different. He saw that the cancer was bringing a lot of basically T cell, a lot of Tregs that are having CTLA4 that are binding to your basically these T cells to stop them, or was actually causing experienced T cells to upregulate CTLA4 to kind of cause basically to kind of stop them basically getting activated from dendritic cells. So they started to realize like, hey, sorry, let me go back in here. They started to realize what happens if we block them. So if we block, for example, CTLA4, we can technically stop this and we can keep getting T cells being much more activated with dendritic cells. We can do that. We can stop this basically inhibitory kind of things, and we can create a huge army of T cells ready to go and kill. Because now you're stopping the CTLA4 kind of activation. Because this CTLA4, again, your tumor's making your T cells upregulate CTLA4 to stop them getting educated from your dendritic cell. Well, let's block it. Now we're letting dendritic cells educate our T cells to come and kill your tumor. But what happens if we also block PD-L1? If we block PD-L1, the T cells that are now ready to come and kill your tumor, they can because they don't have any more this defense system, and they're able to come and recognize the MHC with their peptide. So CTLA4 and PD-1 or PD-L1, they work in different axes. The CTLA4 works mostly in the lymph nodes because this dendritic cell presenting your tumor peptide to your T cell to educate is happening in your lymph node. And this is happening more in the tumor level, where now your T cells that are basically educated, so coming from here, then now they're educated, they're trying to kill the tumor, but the tumor is not allowing it because of the PD-L1. So what you can do is you can use CTLA4 antibodies to help create an army of T cells, and you can use PD-L1 or PD-1, basically either one of them, to block to get these now army to kill. And that's what the new therapy started to come out. So one of the first ones that came out was CTLA4, which basically they called this ipilimumab. Ipilimumab basically, as I said, it stops the CTLA4. It stops CTLA4 from doing this negative inhibition, and it creates a huge army of T cells to go and kill your tumor. And it works very, very well. So this is basically nice examples coming from the clinic. So basically here you have three different, these are two CT scans. This is basically more of a brain CT scan. So you see basically the arrows are pointing to the tumors, and you can see the pre-treatment and then the post-treatment. And you can see from the post-treatment, these people actually had no tumor. People were giving CTLA4. Actually, the people that responded to CTLA4 actually were cured. A lot of them, a lot of people that were responding started to get cured. But the issue with CTLA4 antibodies is that you're stopping your peripheral tolerance. So of course you're getting a lot of inflammation, a lot of T cells starting to kill different organs. So that's why you start getting side effects like dermatitis, colitis, hepatitis, a lot of different inflammations all around. So it comes at a cost. PD-1, PD-1 now really works also in a different, let's say, axis. So CTLA4 again works on creating a huge army. PD-L1 or PD-1, they work more on an axis that you're trying to remove a defense system. So you already have this army, but you're trying to allow this army to kill. So this is the difference between PD-1 and CTLA4. And PD-1 also worked extremely, extremely really well. So again, this is just to show you the differences. CTLA4, you're really working on more getting a huge T cell army. The PD-1 or PD-L1 axis, you're trying to allow this army to kill your tumor cells. Now, the new combinations is to add them both together. Because imagine if you have CTLA4 addition, you get an army of T cells. Great. They come and they're trying to kill your tumor cells, but they're not able to kill them because they have a lot of PD-L1, the tumor cells. Well, you can block it, and now you're allowing this huge army that you created to come and kill. So having CTLA4 and PD-1 together is extremely potent. But this comes at a cost because both of them can increase a lot these immune adverse events. You're stopping literally your peripheral tolerance, and you're causing a lot of inflammation. So you need to be a bit careful because a lot of patients will die from these adverse events. So there's a sweet spot that you need to hit. So if you look at the number of clinical trials, really, they're surpassing the thousands now. It's crazy. And now almost every single cancer, not all of them, but majority of them, are actually having checkpoint inhibitor therapy being used. Another one is basically the final one I want to talk about is immunoviral therapy. Immunoviral therapy is using oncolytic viruses. So oncolytic viruses are very interesting viruses. So usually normal adenovirus infection, what happens? A virus, basically the adenovirus comes in and infects a normal cell and it produces E1B. What E1B does, it stops P53. P53 is a protein found in your normal cells, and P53 stops viruses from replicating. So for adenovirus to keep replicating, it produces E1B to stop P53. And this allows the adenovirus to replicate. What we can technically do is we can actually take an adenovirus and mutate its E1B. So what happens is that the normal cell, when it comes inside, the P53 is able to stop it. And now, in a normal cell, this adenovirus dies. But in a tumor cell, something like 70 to 90% of tumor cells of almost every single cancer, the P53 is mutated. It doesn't work. So now, this oncolytic virus is selective. We've made it conditionally replicate. It will only conditionally replicate in the tumor cell. And now it's able to kill the tumor cell. This is basically selectively replication. And there's many different types of ways to do that. So one of them is basically through this E1B. Another one is basically through this E2F that binds to retinoblastoma. So you're using basically, because tumor cells have different, let's say, signaling pathways or different proteins mutated, you're trying to utilize that. So now this adenovirus is only killing in a tumor cell, but in a normal cell, basically that P53 is actually working, it's stopping it. And this is just an example of basically one of these oncolytic viruses that when you inject, you really get very good, basically killing your tumor to the point that now you basically are. So if you look at these are basically your tumors. They're shrinking them by almost 80%. There was something like here. These are all the tumors. They're again being shrank. So very good tools. And there's a lot of different types of viruses, not just adenoviruses, but you have herpes viruses, reoviruses, measles, and whatnot. But one thing that we started to see that these oncolytic viruses, they work at every single level. They, other than just killing the tumor cells, they can actually cause the dendritic cells to be get activated. They can cause them to go to your lymph node, and they can actually bring T cells and get traffic them much more into your tumor cells. So they work on every single aspect. So this is what we basically, my boss works a lot in and basically optimizing these oncolytic viruses to work in every single aspect. And one of them that I want to really speak about quickly is that what he did is he actually got viruses to dress in tumors' clothing. So basically, he made them into a vaccine. So he actually got, so this is your adenovirus, oncolytic adenovirus. This now, what he did is he actually stuck tumor peptides on top of it. So what would happen is that when you get your dendritic cell to come in, it would cut it into pieces. But the pieces that were basically being cut are mostly the peptides that it basically was covered in it. And then it would teach basically your dendritic cells would teach your T cells to go and kill anything that has this basically tumor peptide, which are basically your cancers. And the T cell will go in and basically kill all of them. And he called this basically pepticrad. So what he did is that the adenovirus is negatively charged. He would select tumor peptides and add a linker. So these are positive amino acids that would basically very linked. And what this tumor now peptide is positive because of this linker. When you mix adenovirus that's negatively charged with this positive linker, literally by just adding them, 15 minutes, they complex. So you actually have an adenovirus that's complex. So this is a very nice versatile tool because all you need to do is just change the tumor epitope. You change whatever epitope you want, and you can change the vaccine. So similar to your mRNA vaccine, now you have adenovirus with different vaccines. So you can make it more personalized. And this is just an example. So what you have this mouse actually having basically the skin cancer that expresses OVA. This is ovalbumin. And basically this OVA has this very specific peptide called symfecol. So this is the tumor peptide. So what he did is he had this oncolytic virus that was able to basically disrupt the tumor. But he basically had the same oncolytic viruses, but then he basically covered it in synfacol, this tumor peptides. So here you have basically the tumor growth, and you see that mice given basically this peptide covered completely in the synfacol or ova, which are the same, they're basically having no tumors at all because they're creating a T cell response that are literally recognizing this synfacol. This actually now is in phase one clinical trials. I wanted to speak about my kind of therapy, but of course I've recorded it and I've added it now here. But this is basically my boss really found a way to get T cells, but he still didn't find a way how to basically tackle this immunosuppressive environment. So what I've done is I've literally created an oncolytic adenovirus, same thing as peptidograd, but this peptidograd would release an antibody that would actually dismantle completely this immunosuppressive barriers, the cells. So now the vaccine would work much, much more better. And this is now the new wave, our new basic company that we're coming out with. So I'm going to stop sharing. Again, guys, this just as there's a lot of examples that you can basically use. I've recorded this also. Now I've recorded this. I'm going to add it to the Blackboard, but I've also pre-recorded it explaining much more. So use this to your advantage. Basically, I will be adding a lot of more papers where you can use them for your essay. If there's any questions, please, guys, let me know. Well, guys, if there's no more questions, thank you so much. I'm going to stop the recording.